Hi, my name is Jay McCurdy. I'm an assistant professor at Mississippi State University and a co-principal investigator working alongside a nationwide group of faculty seeking to understand the ecology and biology of the troublesome turfgrass weed Poa annua, or annual bluegrass. Welcome to Module 2, which covers the fundamentals of herbicide resistance. If you haven't already done so, please make sure to watch the previous module for a more in-depth understanding about annual bluegrass. In this module, we'll discuss how herbicides work, herbicide resistance, and best management practices needed to prevent or overcome the issue. Best management practices, or BMPs, are often discussed out of context with real-world scenarios and practical solutions. Herbicide resistance costs U.S. turf managers money by affecting aesthetics, playability, surface stability, or even by affecting yield, such as is the case for turf or seed producers. In order to control resistant weeds, turf managers may have increased input costs, such as cultural or mechanical means of weed control, or by choosing alternative chemistries when available to them. Effective herbicide resistance management requires a balance of chemical and non-chemical tactics. Best management practices include decreased reliance upon chemical weed control, and increased diversity of traditional cultural practices, such as selection of appropriate turf species for the scenario or environment at hand. The emphasis should be on providing a dense and competitive turf sward. A holistic approach of managing soil and plant health is required in order to do so. When herbicides are used, we must diversify our sites of action between years and within seasons. And we must also emphasize pre-emergence followed by post-emergence means of control, which effectively reduces the amount of annual bluegrass exposed to post-emergence strategies. The objective of this module is to explain the fundamentals of herbicide resistance so that we can better prevent and manage herbicide-resistant annual bluegrass populations. Those fundamentals include how herbicides work, as well as how they are classified according to their site of action, or SOA. How do herbicides work? A herbicide may be deposited on a plant leaf surface, or it may be applied to soil where plant roots come into contact with it. In some instances, a herbicide's activity does not require transport within the plant, and it is considered a contact herbicide. Diquat is a good example of a fast-acting contact herbicide that does not rely upon movement within the plant in order to kill it. Alternatively, in order for some herbicides to work, they may require transport or translocation within the plant. Glyphosate is a good example of a herbicide that relies upon translocation within the plant in order to kill it. Contact herbicides generally work more rapidly than those that are systemic. For example, they may disrupt cell membranes and lead to fluid leakage and rapid degradation. Alternatively, systemic herbicides translocate to the roots or shoots of plants, which can result in better weed control, especially when dealing with difficult to control weeds like annual bluegrass. In many instances, systemic herbicides slowly starve plants by limiting metabolic processes, such as the process of light harvesting or production of amino acids. Herbicides affect metabolic processes occurring within plants. A mode of action describes the broad process within the plant that is disrupted by a herbicide. Another way to think of this is how the herbicide works. For example, an amino acid inhibitor disrupts the formation of amino acids, which are building blocks of proteins. But there may be several different sites or mechanisms by which this is possible. In most instances, herbicides target specific enzymes that are catalysts for reactions or may prevent reactions from occurring. So in other words, they help reaction occur or prevent them from occurring that otherwise would never occur or would occur so slowly as to be impractical to a living organism. Enzymes have specific substrates that must be bound in order to produce a product. Products may include precursors to or even the final products themselves, things such as sugars, fats, and amino acids. They may even include cofactors for other important metabolic processes, such as light harvesting or energy production. Herbicides work by binding to specific enzymatic sites, ultimately leading to buildup of substrates that may be toxic in high concentrations, or by resulting in a toxic byproduct. This precise physiological binding site is used as a way to classify groups of herbicides. It is often referred to as the herbicide's site or mechanism of action. In 1997, the Herbicide Resistance Action Committee, HRAC for short, and Weed Science Society of America, WSSA, approved a herbicide categorization system based on herbicide site of action. The classification system was developed with the idea that if herbicide site of action was well known, recommendations for herbicide resistance management would be easier to understand. In the case of the WSSA categorization system, herbicides with the same site of action are assigned a group number. These numbers are commonly listed on herbicide product labels. The EPA currently recommends that herbicide labels display the group number that identifies the site of action for all active ingredients in the herbicide product. An alternative classification system is that of HRAC, which uses an alphabet-based system to classify site of action. When planning a herbicide program to prevent or mitigate the evolution of herbicide resistance in weeds, rotation among herbicides with different group numbers is necessary between years and within seasons. Here you can see an example of a herbicide label showing a WSSA group number. The active ingredient of Monument 75WG is trifloxysulfuron, which is an acetolactate synthase inhibiting herbicide belonging to WSSA Group 2. This location may vary from product to product. Product labels will also have somewhat boilerplate phrasing about site of action within the text body of the label. Careful consideration of herbicide site of action is an important part of chemical weed control. In some instances, herbicides may be further classified by site of action and even herbicide families. An even further advanced understanding of herbicide action is based upon the herbicide family and potential differences between other families within the same site of action. This classification hierarchy is somewhat useful for understanding how different herbicides have related modes and sites of action. For instance, here you can see that groups 5 and 7 within the broader mode of action of photosynthesis 2 inhibition include various herbicide families and active ingredients that we're familiar with. Group 5 includes the triazine, atrazine, or Atrex, as well as the triazonone metribuzin, or Syncor, and Group 7 includes the substituted urea, Siduron, or Tupersan. While these herbicides may all have the same broad mode of action, they also have slightly different sites of action or binding mechanisms. This figure represents the Herbicide Resistance Action Committee classification of modes and sites of action. There are currently reported cases of resistance in 22 of the 25 known sites of action. There are approximately 160 different herbicides, roughly 50 of which are used in the U.S. turf market. Our next slide will reduce this group into a list of herbicides commonly used to control annual bluegrass. Annual bluegrass has historically been an important weed of many, if not most, commodity and specialty crops. The extensive reliance upon herbicides as the primary means of control has led to almost overwhelming presence of herbicide resistance. There are very few commonly utilized herbicides that annual bluegrass has not evolved resistance to, albeit often in isolated or unique populations. The worrying trend is that for some turf scenarios, we no longer have effective chemical means of controlling annual bluegrass. The sites of action listed in this table represent the currently reported cases of annual bluegrass resistance to various herbicides. The center column contains the WSSA shorthand numbers representing each one of these sites. We'll learn more about these later, but they are meant to help practitioners to quickly and easily distinguish between different sites of action. The active ingredients reported in the column on the right represent relevant active ingredients within the turfgrass industry. Many of us will recognize common active ingredients such as simazine and atrazine or glyphosate, which are standard means of chemically controlling annual bluegrass. You may also be familiar with cases of suspected herbicide resistance to the likes of trifloxysulfuron, the active ingredient in Monument, or 4-amsulfuron, the active ingredient in Revolver. These acetolactate synthase inhibitors are frequently used for control of annual bluegrass in warm season scenarios. How do plants become resistant? Repeated use of herbicides with the same site of action selects for herbicide-resistant plants. In this photo, we observe that only a few annual bluegrass plants are susceptible to the applied post-emergence herbicide. In this case, that herbicide was the acetolactate synthase inhibitor, trifloxysulfuron. Uncontrolled plants reproduce, leading to an increased prevalence of resistance within a local population. That population may move off-site as seed, on equipment, or athletic attire. Controlling these resistant populations can be aided by understanding what herbicides they may be resistant or susceptible to. But ultimately, chemical weed control with multiple sites of action will be more effective than repeatedly relying on the same failed treatment. It is also well known that larger plants, such as those pictured here, are much less susceptible to post-emergence herbicides. Treatments prior to tillering are most effective, and when possible, treatments when temperatures and light conditions are conducive to active growth and herbicide uptake may also aid in control. Mechanisms of resistance are simply how plants are able to tolerate or recover from herbicide application. Normal enzymatic activity serves to produce an end product that is useful to a plant. In this image, you see that a substrate is chemically bound to an enzyme. That enzymatic activity is to convert that substrate into a product. But if herbicides target that substrate's specific binding site, it prevents normal function. This may lead to the inability to produce substances needed by a plant, or it may lead to a backup of toxic byproduct. However it affects the plant, these specific enzymatic binding locations are what we call the target site. Target site resistance occurs when the intended herbicide binding location undergoes a conformational change in structure. This change prevents or reduces the binding of a herbicide. Most populations have very low occurrences of this structural defect. In fact, these binding sites that are structurally different from the normally present in the natural population may have a negative consequence on the plant's health, referred to as a fitness penalty or fitness cost. It is the process of repeated selection using the same herbicide site of action that expands this trait across an entire population, leading to full-blown resistance. Non-target site resistance, sometimes called absorption or translocation-based resistance, may also lead to ineffective control. In this example, a plant may evolve or be selected to have thicker cuticle layers or a novel composition of waxes and fats within that cuticle, such that a particular herbicide treatment no longer is absorbed through the foliage. Again, repeated selection for this trait expands its presence in the population. Another potential mechanism of non-target site resistance occurs when plants evolve methods to mechanically cleave or shed an affected plant structure, such as a leaf, root hair, or tiller. Metabolism-based resistance also occurs. This phenomenon may be due to defense mechanisms intended to protect a plant from foreign substances. For example, increased concentrations of metabolic proteins like cytochrome P450 may bind to herbicides and render them inert or ineffective. Or plants may evolve the ability to sequester herbicides inside the vacuole or other storage structures, rendering them unavailable for action. The Herbicide Resistance Action Committee has issued somewhat broad best management practices. In truth, no single practice is going to completely eliminate annual bluegrass from turf. But certainly the most valid advice is to have an integrated approach. This means using multiple sites of action with overlapping weed spectrums in rotation between years or seasons. as well as in sequence within season and mixtures at individual application timings. Cleaning equipment, albeit imperfect, is necessary to prevent trafficking of seed from resistant populations. Furthermore, a common source of contamination is due to seed dispersal on athletic attire, such as golf shoes. In the same spirit, managers may also quarantine populations where resistance has been confirmed. Using full labeled rates at proper timings has always been an important means of successful weed control. But too often, practitioners skimp on required rates in order to reduce costs. When resistance is considered the best weed control option is often the most optimal in order to prevent populations from reproducing. Finally, scouting for escaped weeds after a herbicide application is often necessary. And when possible, eradicating escapees is the best means to prevent weeds from going to seed or proliferating vegetatively. Finally, these authors, as well as team members within their labs, are to be given credit for their development and review of this module. Financial support for this work was provided by the United States Department of Agriculture Specialty Crop Research Grant. Our thanks are extended to the Weed Science Society of America and Herbicide Resistance Action Committee for allowing us to use training materials posted on their website as the starting point for these lessons. Again, I'm Jay McCurdy. My colleagues and I welcome your input and participation. Thank you.